One of the most fascinating aspects of science is, however esoteric and of less applicability an area might be, we never know what happens in the future. This is especially the case of my field which is protein folding. We apply this nowadays to two different diseases. One is cancer, and also separately in neurodegenerative diseases. We look at the molecular level details of how a polypeptide chain folds and gives forth bioactivity. One of the ways that we observe this is to use a instrumentation called NMR spectroscopy. NMR stands for nuclear magnetic resonance, which is a technique that we use. We have an 800 megahertz or an 18.8 Tesla spectrometer that is able to look at molecules, biomolecules, inorganic molecules, all kinds of different atoms and molecules at atomic resolution. And we're able to extract the information that we need from various pulse sequences, various experiments. So our lab does more or less three different types of experiments. One is the methodology development of biomolecular NMR spectroscopy. And to this end, we are looking at partially unfolded proteins that are very intractable to observe with other methods. We also want to develop methods for biometabolomics, looking at the various metabolites within our sera or urine and so on, and trying to diagnose what a patient is. This is especially relevant because we have a wealth of wonderful biobanks here in Ireland, and we are able to look at many of these samples, these thousands and thousands of different samples, and trying to characterize them and relate them to one another. Our second aspect of experimentation is on this partially unfolded protein fatty acid complex, which is nicknamed Hamlet, which stands for human alpha-lactalbumin made lethal to tumor cells, HAMLET, a invention that was done by our close collaborators in Lund University in Sweden, where we look at the structure and how it actually can selectively target tumor cell lines and rather keep the healthy differentiated cells intact. It selectively kills cancer cells. That's one of our great interests in how this can go on. We also are able to see that it doesn't have to be this particular protein. It can be other proteins. So there is more of a generality to there. Our third and final project that we are very interested in is designing peptidomimetics against various amyloid fibrils in the hopes that we are able to inhibit fibrillogenesis, which obviously it has a strong correlation with various neurodegenerative diseases like Parkinson's, Huntington's, and Alzheimer's diseases. One of the offshoots that we've got some excitement in is when we are not successful in designing an inhibitor, we are yet successful in getting some amyloid fibrils that have interesting bio-nanotechnological properties. And we are trying to exploit those properties, hopefully to get some very nano-sized light harvesting complexes or other type of applications. And that's more or less what we do. So our area of interest is in protein folding and the applications to cancer and neurodegenerative diseases. The technique or the instrumentation that we use is NMR spectroscopy. The spectrometer we currently manage is the largest magnetic field on the whole of this island. So we have various collaborators from all around the Republic of Ireland and Northern Ireland as well. And the field that we're interested in is in protein folding and misfolding. Thank you very much.